So thank you, Carlos. So the next speaker for today is Dr. Naveed Akbar. Naveed, you can start sharing your screen. So Dr. Akbar obtained his PhD in medicine and therapeutics at the University of Dundee. During this time, he investigated the role of innate immune signaling cascades in vascular inflammation using in vivo imaging. Dr. Akbar is currently an associate professor of cardiovascular science at the University of Oxford, and his lab is interested in defining the therapeutic and diagnostic potential of extracellular vesicles for immunomodulation and precision medicine in cardiovascular, metabolic, and inflammatory diseases. So today, Dr. Akbar will talk about the interplay between EVs, immune cells, and inflammatory responses in the context of acute myocardial infarction. So Naveed, the floor is yours. Thank you very much.
Thank you very much for that. Yeah, sorry, I couldn't unmute myself, but thank you very much for the introduction, and thank you everyone for the opportunity to share our work. So I've recently established my lab in the Division of Cardiovascular Medicine at Oxford, which focuses on studies from my postdoc. So I have an interest in how endothelial cell-derived extracellular vesicles can drive disordered innate immune signaling to contribute to cardiovascular disease. So today I'll share with you some data from published studies and analyses that we have taken in patients experiencing a heart attack. So acute myocardial infarction is an important pathology within its own right, but it's also a model of sterile inflammation. So a rupture or a blockage within an atherosclerotic plaque within the heart results in a heart attack, where you get this localized ischemic damage and injury to the myocardium. The primary therapy here is percutaneous coronary intervention, but this doesn't always result in better outcome for patients. And by that, I mean longevity and healing of the heart in all the patients. And that's because we have an inability to recognize which patients are going to do well and which patients might need some additional focused therapy. And this is further amplified in patients with metabolic disease, where some patients are more likely to have a larger heart attack, and they're more likely to have poorer healing as well. We are also limited by the number of methods and treatment options to help the heart heal, but targeting the immune system might be one such possibility. So we see a very strong and a coordinated immune response following injury to the heart. Within the first few hours, millions of neutrophils enter the peripheral blood. And from here, they're able to infiltrate the ischemic myocardium. And neutrophils are really volatile cells. So once they enter the myocardium, they undergo a process of degranulation, where they release pro-inflammatory toxins and proteins, and they prime the environment for the subsequent arrival of monocytes. Monocytes are really well studied within the setting of heart injury and repair, and they contribute both to the pro-inflammatory injury that we see within the first couple of days after a heart attack, but they also contribute to the repair and the regeneration in the subsequent days and weeks. So these very rapid elevations in neutrophil number and in monocyte number in patients, they're prognostic. So they determine the size of injury to the heart. So more neutrophils or more monocytes in the blood of the patients as they arrive at the hospital, it results in a larger myocardial infarction, a larger scar size. And that's also associated with a high incidence of mortality. So we have applied transcriptomic techniques to these mobilized peripheral blood cells, and we've shown that they're transcriptionally activated and they're favoring pathways for inflammation, for proliferation, before they reach the injured myocardium. And this changes the way that we think of immune cells. They're not just responding to the local microenvironmental cues within the ischemic heart, but they're showing some level of priming or programming. And we think that's really exciting because it opens the possibility then of targeting these peripheral blood immune cells for therapeutic immunomodulation before they get to the injured heart, possibly allowing us to push them towards repair and regeneration and away from their pro-inflammatory function. So the question arose, how are these cells both mobilized and transcriptionally activated? In a series of experiments, I've been able to show that following injury to the heart, you get enrichment of endothelial cell-derived extracellular vesicles, which carry this VCAM1 protein on the surface, and they're also enriched for the endothelial cell-associated microRNA 126. These can very rapidly localize to the spleen, and they're able to mobilize both neutrophils and monocytes, and they can induce their transcriptional activation. So over the next 25 minutes or so, I'll share some of these data with you. So we have a fantastic study in Oxford called the Oxford Acute Myocardial Study, where we recruit patients who are presenting with a heart attack, and then we can undertake in-depth phenotyping with patient-specific MRI and repeated blood sampling. In agreement with previous studies, we find that the plasma neutrophil number at the time of presentation, this is when the patient arrives at hospital, correlates with the extent of myocardial injury. So in these graphs, you can see the number of blood neutrophils from the patients when they arrive at hospital, which is on the y-axis. The extent of injury is a measurement of edema by T2-weighted MRI, and late gadolinium enhancement is used 6 months after the initial heart attack to determine the size of the scar. In both measurements, which are the same patients, we find that more neutrophils in the peripheral blood as the patient arrives at hospital, it results in a larger infarct. This very rapid elevation in peripheral blood neutrophil number following the heart attack raises a question. How are the neutrophils mobilized, and where are they possibly mobilized from? And it raises that question because it's far too quick for more complicated processes such as proliferation or myelopoiesis. So to determine whether the spleen or the bone marrow mobilize neutrophils after injury to the heart, because these are reserves of cells, I used a mouse model of acute myocardial infarction, where we take an otherwise healthy mouse. We're able to insert a suture within the region of the LAD. Then we allow the animal to recover, and at set time points, we harvest tissues of interest, such as the blood, the spleen, the bone marrow. And here we've used flow cytometry to look at neutrophil populations. And what we find is that there's a very rapid increase in peripheral blood neutrophil number and a simultaneous reduction in splenic neutrophil number. But at this very early time point, there's no alterations within the bone marrow or within the heart. So this was really exciting for us. Neutrophils mobilized from the spleen following a heart attack. It's not been demonstrated before in sterile injury. And we think that's because traditionally research groups like ours who are interested in rapid immune cell activation and kinetics after a heart attack don't always look at these very early time points. So we hypothesized that the mobilized peripheral blood neutrophils would undergo alterations in their gene expression before recruitment to the heart, similar to what we have previously reported in our patients and in our animal models for peripheral blood monocytes. To determine this, I employed RNA sequencing, where I looked at 20,000 genes at the same time. Here we're using N-STEMI patients as a control, and then STEMI patients at the time of presentation when they arrive at hospital, and then we bring the same patient back 1 month later to serve as a matched control. So using this type of unbiased analysis in this PCA plot, we find that STEMI patients are grouping together, but there's a very clear delineation between the response at the time of injury when they're coming into hospital and then the follow-up time point 1 month later. If we look more closely at the absolute number of genes that change within each group, which are highlighted in red, we find that over 900 genes are changing in STEMI patients, which is surprising because neutrophils are regarded as cells which are transcriptionally poor. But what are the cells doing? So employing further bioinformatics here, we use a technique called gene set enrichment analysis to determine whether the genes that are changing are favoring any particular pathways. And we find that there's strong representation of inflammatory pathways with significant enrichment for pro-inflammatory, TNF-alpha, and interleukin-6, which isn't surprising for a neutrophil, but the fact that these neutrophils are transcriptionally switched on before they get to the heart, this is very exciting for us. So if immune cells are programmed or primed before they get to the site of action, then we can possibly therapeutically manipulate this en route before they get to the heart. So neutrophils are mobilized from the spleen very quickly following a heart attack, and they show transcriptional activation within patients. But how is this response coordinated? So chemokines are not elevated at this very early time point, and the concentration would possibly be inverted. It might be very high within the myocardium, and it might be really low in the peripheral organs, such as the spleen. So we hypothesize that extracellular vesicles, which are membrane-enclosed particles, may mediate this response, similar to what I've previously shown for splenic monocyte mobilization. So these are some schematics from a review we published a couple of years ago, showing just the three major populations. But the data I'll share with you over the next few slides is data derived from ultracentrifugation. We know very well now that the method employed can influence the type of EV you isolate, and different methods carry varying levels of these so-called contaminants. So in plasma EV preparations, this includes lipoproteins and soluble proteins such as albumin. I'll share with you towards the end of my talk another study where we've undertaken a systematic analysis of 5 different EV isolation methods, both in healthy volunteers and in patients following acute myocardial infarction. So I tested the hypothesis that plasma extracellular vesicle number is altered following myocardial infarction, and that this may drive the neutrophil responses that we're observing in our patients. So we use the Oxami cohort here, and I determined plasma EV concentration using nanoparticle tracking analysis. So this is the Zeta View machine, which we have in our labs. So the concentration of the plasma EVs is on the y-axis, and the size of the particle is on the x-axis. So patients presenting with a myocardial infarction have significantly more particles at the time of injury versus a 6-month follow-up control. But we find that the overall size profiles are similar, with a possible increase within the small EV range. We can look at these isolated extracellular vesicles in greater detail. So here I'm showing you a standard transmission electron microscopy, seeing this typical cup-shaped morphology. And then we're able to blot for extracellular vesicle-associated proteins such as CD9 and CD63. But do these concentrations track with the extent of myocardial injury? So plasma EV concentration at the time of presentation, so when the patient arrives at hospital, correlates with myocardial injury, both within the first 48 hours and 6 months later with the size of the scar. So these are the same patients that I showed you earlier. Greater EV counts corresponds to a larger infarct by both the edema quantification and by the late gadolinium enhancement. What we also find is that there's a really highly significant relationship between plasma extracellular vesicle number and plasma neutrophil number. So this is just an association, but it raises the question that extracellular vesicles may mobilize or interact with neutrophils following myocardial injury. But simply counting particles which have isolated using ultracentrifugation and using nanoparticle tracking analysis isn't enough. Do the extracellular vesicles carry classical markers? Can we quantify them with the very limited amount of clinical sample that we have? And are we able to determine which cells are producing them? So our study in Oxford enables us to obtain repeated blood samples from the same patients. So from the moment that they arrive at hospital and over the next 48 hours, and then we obtain a 6-month control sample as well. To allow quantitative analysis, I've used here a technique called the EV array, which is collaborators from Orborg, which binds extracellular vesicles to the array surface using a combination of CD9, CD63, and CD81. And what we can do subsequently is probe for markers that we're interested in. So using the array, we initially looked at the EV response using extracellular vesicle-associated proteins such as the CD9, CD63, CD81, and ALX TSG. And what we find here is that there's a very rapid elevation in circulating plasma extracellular vesicles as soon as the patient arrives at hospital, so prior to clinical therapy. But there's many cell types within the heart that can produce extracellular vesicles, And so we hypothesized that it might be venial endothelial cells. So these cells are in very close proximity to the ischemic area. They're kind of bathed in with the ischemic environment, and that might be an area which might generate a signal to the periphery for immune cell recruitment. So in the same patients, we looked at the same time points and had a look at endothelial cell markers and other markers, including tissue factor and the immune cell marker CD16. So the endothelial cell activation marker VCAM1 is the most enriched at this time point as the patients arrive at hospital. But prior to this, VCAM1 had not been associated with the surface of extracellular vesicles. So to determine that VCAM1 was present on the plasma EV extracellular vesicle surface, I isolated some plasma extracellular vesicles using ultracentrifugation, and then I took these magnetic beads and conjugated anti-VCAM1 antibodies to the surface. What we can then do is we can incubate these beads with our isolated sample. You can then embed them within agar, you can section them, and you can look at the surface of the bead. And what we find is that the bead surface is decorated with VCAM1 positive extracellular vesicles. So I'm not showing you the IgG beads here, but they're in the published manuscript, and they're completely clear. So this accumulating data was suggesting to us that endothelial cell-derived extracellular vesicles, possibly rich in VCAM1, might be signaling neutrophil mobilization following a heart attack. However, it's really difficult to determine the biodistribution of extracellular vesicles in vivo because they're very, very small. But to enable us to look at this, I developed a labeling technique that would allow me to quantitatively assess extracellular vesicle biodistribution in the mouse over time. So we used an established model of endothelial cell activation. So here we're using mouse endothelial cells. And what we know is that they produce and release extracellular vesicles under normal basal conditions. And we can model heart injury by using the pro-inflammatory factor TNF-alpha. And when we do this, we see almost a doubling in the number of particles released from extracellular from endothelial cells. And you can also see here a traditional TEM of these kind of cup-shaped morphologies, which we know is a dehydration artifact of extracellular vesicles. But if you look at the same sample using cryo-TEM, you can then see their native membranes and possibly their structure of what it would be like in an aqueous buffer. So these cell culture derived endothelial cell derived EVs, if you stimulate them with the pro-inflammatory TNF-alpha, we also find that there's an enrichment for VCAM1 on the surface, similar to what we find in our clinical cohorts. To allow quantitative tracing of these endothelial cell derived EVs from the mouse, we took a preparation and I transfected them with a non-mammalian microRNA MER39. So this isn't present in the mouse. And then what we can do is we can wash them and we can inject them by tail vein into the mouse. We can then take tissues of interest at various time points, and we can use RT-qPCR to see where they end up and how long they remain detectable for. And what we find is that they're delivered to the spleen within several minutes, and they remain detectable for up to 24 hours. Now, there are some inherent issues with this technique. Is it still the EV that you're detecting, or is it just the C. elegans microRNA that we're detecting at the later time points? But it does allow us to determine that they're delivered to the spleen very rapidly within a couple of minutes. So extracellular vesicles decorated in VCAM1, localized to the spleen, but do they orchestrate this neutrophil mobilization response that we observed within our clinical cohorts and then within our rodent model? To test this, I took otherwise completely healthy mice. I injected them with the preparation of inflammatory activated endothelial cell derived EVs, harvested tissues of interest, and looked at neutrophil populations using flow cytometry. So the amount I'm injecting here represents less than 1% of the endogenous circulating pool. What we find is that there's a very rapid and substantial increase in peripheral blood neutrophil number, and we get a simultaneous reduction in splenic neutrophil number, but no alterations in the bone marrow. What we were also able to do is we followed up these animals and we looked at whether any chemokines were changing within the spleen, which might mechanistically allow us to look at what is allowing the neutrophils to deploy from the spleen. And one of these that was of interest to us was a protein called SDF1. So we looked at 20 chemokines initially using RT-qPCR. We find a number of alterations, but then if we look at the protein concentration, we find that a number of those are changing as well, in particular, stromal-derived factor 1. So ordinarily, the expression of SDF1 is quite high in the bone marrow in the spleen, and it retains neutrophils to their reserves. And so what we're seeing here is a downregulation, and you can achieve this as well by just giving an antibody against SDF1. And then what you notice is you get neutrophil mobilization to the peripheral blood. So these experiments support the hypothesis that endothelial cell-derived extracellular vesicles mediate neutrophil mobilization from the spleen with localized chemokine signaling within the spleen itself. But what mediates the effect? What's important about the extracellular vesicle? So it's really well known that immune cells can be captured to endothelial cells through interaction with VCAM1 and the receptor VLA4, which is found on neutrophils and on monocytes. We find that VCAM1 is enriched on plasma extracellular vesicles in our patients and on the endothelial cell-derived EVs from culture. So we hypothesize that the inverse might be true, where plasma-liberated extracellular vesicles enriched for the VCAM1 could localize to the spleen and then mediate neutrophil mobilization. So to test this hypothesis, I employed CRISPR base editing. We introduced a stop codon within the VCAM1 gene in mouse endothelial cells. So if you apply the pro-inflammatory TNF-alpha, you don't express VCAM1, unlike in the wild-type cells, which you can see here on this Western blot. I then generated these wild-type VCAM1-rich endothelial cell-derived EVs and those that were deficient. But you can still see that they're carrying these standard extracellular vesicle markers, such as CD9 and TSG101. So we hypothesized that the VCAM1 may be necessary for delivery of endothelial cell-derived EVs to the spleen, for the functional effects, such as the alterations in gene expression, for the protein changes that we've seen, and therefore driving the mobilization of the neutrophils. So to test this, I took wild-type mice again, completely healthy, injected them with the wild type of the VCAM1 knockout extracellular vesicles at the same concentration. And then 2 hours later, following the injection, we take tissues of interest and we're able to look at the neutrophil populations using flow cytometry. So we find that the wild type extracellular vesicles mobilize neutrophils to peripheral blood, but this is blunted within the VCAM1 knockouts. Thereby, we're demonstrating a pathway where ischemic tissues liberate extracellular vesicles from endothelial cells to mediate chemokine regulation and mobilization of neutrophils from the spleen following acute myocardial infarction. So I won't go into these additional data here, but we show that the chemokine differences that we were observing with the wild-type extracellular vesicles are also blunted if we remove the VCAM1. So plasma EVs contain a number of bioactive molecules, including RNAs, and amongst the various RNAs, microRNAs are really well studied, and they can mediate gene expression changes within the recipient cells. And I've already shared with you some data on how neutrophil transcriptomes are altered in patients as they arrive at hospital. So we've got evidence that extracellular vesicles decorated in VCAM1 can drive the mobilization of splenic neutrophils, but might they be also leading to a priming or this programming effect that we're seeing within the patients? So to determine this, we isolated plasma extracellular vesicles. We used the focused plasma microRNA array, where we looked at over 700 different microRNAs using RT-qPCR. And what we find within our patient cohort at the time of presentation, there's 12 microRNAs which are significantly altered. Now, there's many cell types within the heart that may generate extracellular vesicles, and indeed extracellular vesicles that have changes within their microRNA profile. But all of these microRNAs are present within endothelial cells, And then two of these, microRNA 1263P and microRNA 1265P, are considered endothelial cell associated or endothelial cell specific. So this was adding some weight to our building narrative that plasma extracellular vesicles are originating from endothelial cells. They might be decorated in this endothelial cell glycoprotein. They can localize to the spleen, they can mobilize the cells, and that their contents then, which they contain, these microRNAs, might contribute to the priming or the programming effect that we're seeing within the peripheral blood neutrophils. So we're a translational lab, and we like to use our clinical cohorts to hypothesize for subsequent investigations in vivo. So the model that I showed you earlier of where we're using these mouse endothelial cells, we can do an equivalent using human endothelial cells. So here we're using HUVEX. We can take them under control conditions, and we can stimulate them with a pro-inflammatory TNF-alpha. We can take the supernatant and then isolate the extracellular vesicles using ultracentrifugation in a similar way. So the human endothelial cells also produce extracellular vesicles under basal conditions. And when we apply the pro-inflammatory TNF-alpha, we get this doubling response, which appears to be conserved between the mouse and the human. We can have a look at them using transmission electron microscopy and cryo-TEM. We've done the assessment of their extracellular vesicle markers using Western blot. But what we also find is that there's enrichment for the microRNA 126 within the extracellular vesicles that come from the inflammatory activated cells. And this supports or confirms our clinical observations that maybe activated endothelial cells release more extracellular vesicles, rich in VCAM1 on the surface, but also rich within the microRNA 1263P and 5P. So this model allows us to look at these complications of endothelial cell activation following myocardial infarction. So microRNA 126 is enriched within these endothelial cell-derived extracellular vesicles, but does it have any relationship to neutrophil biology? And how do we know that it might be governing that neutrophil response that we're seeing after myocardial infarction? MicroRNAs have this putative seed sequence, which allows us to look up in curated databases their predicted of putative targets. So here we've used a number of different targets. I used 3 different databases to enhance kind of the stringency of my analysis. So to determine whether microRNA 126 targets are enriched within neutrophils following a heart attack, we looked at the STEMI patients that I showed you earlier, where those 900 genes are changing. And I compared the putative or the predicted list of microRNA 126 targets to the genes that are actually changing. And what we find is that there's a significant overrepresentation of these predicted targets in those genes that are changing using a Fisher's exact test. But I wanted to follow up on these studies within our mouse model, so where we can test whether therapeutic interference with this signaling pathway might be beneficial. But at that time, it was actually the pandemic, and I wasn't able to approach the investigations myself. But across my neutrophil reading, I came across a preprint by a group in Germany, by Clement's team. And so I reached out to them, who had a rich data set on neutrophil data at a single cell level following myocardial infarction in the mouse. And so using a similar comparison technique, here we can take the predicted targets for the mouse of microRNA 126, and we can say, do those gene targets change within neutrophils following myocardial infarction? And within specific clusters of this single cell data, we find significant enrichment or overrepresentation. So I'm not going to go into the data right now, but it's in the published manuscript. We also wanted to know whether inhibiting the microRNA 126 would be beneficial or whether it would affect infarct size. So we're able to give an antagomer to the mouse prior to myocardial infarction, and then we find that there's a significant reduction within that injury size within the mouse. And we did those studies in collaboration with Catherine Moore's group in New York. I'm conscious of time, so I think I'll move to a summary. So in summary, we find that acute myocardial infarction induces a peripheral neutrophil response that correlates with the extent of myocardial injury in patients. Plasma extracellular vesicle number is also elevated in patients as they arrive at hospital. It also correlates with the extent of injury to the heart and the size of the scar. There's also a relationship between the number of neutrophils in the peripheral blood and the number of circulating extracellular vesicles. Some of these circulating extracellular vesicles show enrichment for endothelial cell origin. In particular, we find this enrichment for VCAM1 on the surface of extracellular vesicles. We know that endothelial cell-derived extracellular vesicles can localize to the spleen very, very quickly. Once they localize there, they can induce alterations in gene expression and protein expression. They can then lead to the mobilization of splenic neutrophils. And we've shown that that is dependent on the surface expression of VCAM1. We've also shown that neutrophils are activated before they get to the site of injury. They show enrichment for microRNA 126 targets, which are enriched within the plasma EVs of our patients when they arrive at hospital, but also within our inflammatory activated cells in culture. And we think that therapeutically targeting neutrophil transcriptomes might be a way in which we can modulate the pro-inflammatory function of these cells to help salvage the myocardium within the first few hours following myocardial infarction. I was going to share some other slides with you, but I will just move across to my acknowledgements instead. So first of all, I'd like to say a big thank you to our funders, the British Heart Foundation, the Oxford BHF CRE, who have supported me with several grants for equipment and travel. A big thank you to all my collaborators and everyone in the lab, including the Oxami study, and the big thank you to the SNEV network as well. So you've invited us a couple of times to share our work. And that's really much appreciated. So thank you for your attention, and I look forward to your questions. Sarah, I think you're muted.
Can you speak?
Oh, yeah, sorry. Oh, okay. Yeah, yeah, again, sorry. Thank you so much, Naveed, for your talk. It was really nice. And I see that Sarah, the other Sarah, has a question for you. So, Sarah, yeah, you can ask it.
Hi, Naveed. Thank you so much. The talk was great. It is a lot of data. It is quite hard to follow up, but I have two questions actually. I'm not entirely sure if you actually answered them or if I just missed it. One of my questions is the circulation. So the neutrophils that are activated in circulation, are they activated in the spleen by the EVs and then released to circulation, or are the increase of EVs in circulation that activate the neutrophils that are not activated in circulation? So what is the case?
Hi, Sarah. Great to see you, first of all. It's a great question. So we don't know the full intricacy of that. We know that extracellular vesicles are delivered to the spleen and that they can interact with the neutrophils and they can mobilize them. And we also know that the neutrophils within the peripheral blood are transcriptionally activated. What we don't fully know, and we can't dissect out in vivo, is where does that programming take place? Does it happen in the spleen immediately, or does some of it happen within the peripheral blood? I would hypothesize that there would be possibly a two-way interaction. Some extracellular vesicles are causing the mobilization, and maybe as they enter the peripheral blood, they then interact with other extracellular vesicles that might cause the transcriptional programming. What's really interesting about immune cells in the peripheral blood after a heart attack is that they remain in circulation for a few hours before they go to the heart. And so the neutrophils deploy almost immediately, but there's a bit of a lag between the numbers increasing in peripheral blood and then entering the myocardium. And I think that is the transcriptional priming or programming window of possibly where extracellular vesicles are exerting their effect.
Right, thank you. I see that Carlos has a question. If I can ask another question and then go to Carlos, it make it easier. So my next question is, you talked about the CTF1 protein, and you said it was highly expressed in bone marrow and in spleen. However, you only see a decrease on the spleen, and while the bone marrow was the same. Do you have any idea why that happens, or did you look into it, or not really?
So the stromal-derived factor 1 is expressed both in the bone marrow and within the spleen. What we know from our investigations, from my studies, is that I think more extracellular vesicles are being cleared by the spleen than by the bone marrow, or what's accumulating in the bone marrow in the time points that we've looked. We know that stromal-derived factor is very important in neutrophil mobilization from the bone marrow. But what we're seeing is that there's more of a contribution within the spleen in these early time points.
Great, thank you so much. It was really, really good talk.
Yes, thank you. So I think now Carlos has another question. So Carlos, you can unmute.
Hi, so thank you so much. Amazing talk, a lot of techniques and a lot of tools to validate your point. I think it's amazing. And especially I really like that you didn't focus on the cardiomyocytes and you look to the endothelial cells, because a few years ago, a few years ago, the focus was always the cardiomyocytes. And now it's really nice seeing that a lot of science is being done in other type of cells. My question is, so it's really nice that you look, so the bridge between the heart and the spleen, but I would say, do you think that other organs could be also involved in this systemic issue? Because the acute myocardial infarction is a systemic issue, it will affect a lot of organs. So I'm popping here, maybe the lungs or even the kidneys, because there is a big direct relationship between them.
Thank you very much for your question, Carlos, and thank you very much for the invitation specifically to you. You're the one who emailed me. It's a great question, because what we already know is within acute injury, and so acute injury could be stroke, it could be myocardial infarction, you see activation or inflammatory processes switching on in other organs that aren't the heart. In particular, renal dysfunction is a complication that happens in patients. We have shown that if you perform an experimental myocardial infarction in the mouse, you can get activation of endothelial cells within the kidney. What we've also shown is that you can have injury to the brain and to the heart, and you get activation of inflammatory pathways within the liver. And we've followed up on some of these liver studies now, where we're finding that the VCAM1 positive extracellular vesicles are also driving this response. And so by removing that VCAM1, we can abolish some of these, what we call APR, the acute phase response.
Yeah, sounds good, sounds interesting. If I may, can I ask another question, Sarah?
Yeah, sure.
So my other question is that at the very beginning, you showed a clear increase of the EVs that are being collected in the plasma of these patients when they are admitted at the emergency room, I can imagine. My question is, because I can imagine that different patients are going to arrive to the hospital with different times since the infarction started, or since the symptoms appear. Do you have any kind of normalization between these patients?
Great question, Carlos. So we do take this into account. So as the patients arrive, or even prior to that, they're asked, when did your symptoms start? And so within all the data that I've showed you today, the median time from chest pain to presentation is 3 hours. And so the studies that I've shown you here are up to about 15 to 20 patients. And so we can plot that, but then it breaks down into quite small groups. So we lose a lot of power there.
Yeah, but between inside, I mean, less than 3 hours is quite amazing. Congrats.
It's a critical time point, right? So we know that the first few hours are the most critical for reperfusion therapy. Reperfusion is going to be effective in managing the heart attack for that patient. So it has to be within those first 4 hours.
Yeah. Thank you so much.
I see that there is a question from Shikra, so maybe a very quick one.
Yeah, sorry, it's going to be super quick. Naveed, thank you so much. First of all, it was a very clear presentation and the techniques were well explained. I had a quick question regarding the localization of EVs within the spleen. Did you happen to look at any other organs or look at a biodistribution, whether they go to somewhere else as well, or it's specifically targeting only the spleen?
Thank you very much for your question, Shipra. So yes, so if you go to the published studies, it unfortunately ended up in a supplementary, and it was a lot of work. And so as we inject the extracellular vesicles into the mice at various time points, we took a lot of organs. So we took the lungs, we took the heart, the liver, the spleen, bone marrow, skeletal muscle. And what we find is actually that the spleen was accumulating the most number of extracellular vesicles as a proportion of tissue relative to the injection itself. So maybe it's a phenomenon that is specific to endothelial cells, derived EVs, rich in VCAM1. I don't think that would be known for all extracellular vesicles coming from all cells or tissues. You're muted.
Sorry, yeah, specifically when we know that EVs tend to go like to liver and, you know, like have this liver accumulations. I was just curious if you saw the highest in spleen, but yeah, thank you.
We do, but the liver is much larger, right? So you have to express it as a proportion of tissue.
Right. Makes sense, yeah.
Thank you again. I have to close the Q&A session. I have a